All right. Thank you for joining us for today's colloquium. I have the honor of introducing today's speaker, Tanner Robertson. Tanner is a postdoc here in Anna Hutmacher's lab. But let me tell you how he got to that point. So he began his research career at Texas Christian University. From there, went to Penn to work on his PhD in 20. You finished in 2021?
Correct, yes.
Penn. So that's where he began working on T-cell trafficking. So, and he continued this investigation of T-cell migration and trafficking here as a postdoc in Anna's lab ever since. One of the fascinating things is that he's working on it in Robertson. And most of us are accustomed to thinking about adaptive immunity in mammalian systems. And when you think about this, the lymph nodes are sort of a cornerstone of the lymphatic system in mammals. They serve as a kind of one-stop shop for T-cell antigen scanning. And you move to a fish, and there's no lymph node. So how does it work in a fish? And so Tanner has done some amazing work on elucidating what a lymphatic network looks like in an organism that doesn't have lymph nodes, and how do the T cells get to where they need to be inside this complex network. And I think that's what you're going to share with us today.
Correct.
So I'm really excited to hear about that. And I also want to point out that something that I looked on a scope with you here today, that Tanner's work in this area has even garnered him a prize from the Cool Science contest. Which is a kind of beautiful, like country to allow us to visualize the lymphatic system. So will you be showing us that image today?
It's actually that fish that's right there, but they blew it up to about the size of a person. It was pretty cool.
Well, congratulations on
Thanks.
that work. It's fantastic work. Work's already as one publication in PNAS and another one it's about to come out.
It's the review has been sent in. We're waiting for them to say yes or no.
So I'm very excited. With no further ado, let's hear from Tanner.
Perfect. Thank you for that introduction. I'm going to be talking about how T cells find antigen in animals without lymph nodes. So a little bit of background about T cells and antigen search in mammals. So our bodies maintain billions and billions of T cells, but each one of these T cells has a unique antigen receptor specificity. Let me get my laser pointer up here. And what that means in the context of an infection is that for any given pathogen or for any given antigen, the frequency of a pathogen-specific naive T cell is going to be extremely rare. On the order of about 1 in a million T cells is going to be specific or useful for any given infection. So this creates a sort of needle in the haystack problem in adaptive immunity. And this is a problem that is made worse by the fact that not only are antigen-specific T cells in the naive pool going to be extremely rare, they also have no way of predicting where in the body that antigen is ultimately going to arise. You can get an infection in your throat, in your gut, in your intestines, in your brain, anywhere. And no matter where that peptide ends up, these T cells need to be able to find it. So the way that mammals have more or less solved this problem is through the evolution of lymph nodes. And what lymph nodes ensure is that no matter where an infection is taking place in your body, peptides from that pathogen are going to wind up in a nearby draining lymph node. You've got an infection in your throat, you've got regional lymph nodes that drain from there. Same thing about in your skin, your gut, your brain, everywhere. So if you're a naive T cell, you don't have to manually search every square inch of the body. All you really have to do is move into and out of these lymph nodes, searching within that lymph node, and then moving on to the next one. This dramatically reduces the amount of space that these T cells have to search. And the reason why we care about this is that naive T cells detecting antigen is really the first step in the initiation of adaptive immune response. And if you don't get naive T cells detecting antigen, you're not going to get the generation of neutralizing antibodies by B cells, and you're not going to get the elimination of infected cells and ultimately the eradication of these pathogens. So this lymph node system works remarkably well, but there's one sort of glaring problem with it from the context of evolution, which is that all jawed vertebrates have diverse T cells, but lymph nodes are more or less exclusive to mammals. You've got some proto-lymph node-like structures in some birds, But more or less, if you are a T cell in a fish or an amphibian or a reptile or most birds, you've got this same fundamental problem of you need to be able to find antigen no matter where it's occurring in the body, but you're missing this critical lymph node infrastructure that's facilitating that search in your mammalian counterparts. So this project really started with a basic question in immunology, which is how do T cells search for antigen in animals without lymph nodes? I actually was a former mouse person. I did my PhD on mice, studying lymph nodes at Penn, and I decided to make the switch to studying zebrafish to try to answer this question. So fish do not have lymph nodes, but a little bit more background. They do have a different kind of lymphoid tissue called mucosal-associated lymphoid tissues. So we also have these. The famous example in humans is going to be Peyer's patches. These are going to be organized groupings of lymphocytes around our mucosal surfaces. So fish have these really beautiful organizations of T cells shown here in magenta around their gills, around their nasopharynx, and to a lesser extent around their gut. And they also have a spleen, which has lymphocytes in it. I'll talk a little bit later about the spleen and why I don't think the spleen is particularly relevant in fish. But because our spleen is important for immune function and fish have a spleen, a lot of the early work has been done on the spleen. So before I decided to do my PhD in this, I originally thought that these lymphoid tissues were probably sufficient for these fish to survive. But the thing that got me thinking that there had to be something else going on was not any experiment that was done in a laboratory, but it's something that people do on fish farms every single day, which is that they vaccinate their fish. And there's a number of different ways that you can vaccinate a fish. The most common ways are going to be immersion or incorporation into feed. So you mix the antigen into the water at a high concentration, or you mix it into the feed. These make sense to me because if you've got a fish that's got lymphocytes around the gut or around the gills, it makes sense to me that they would be able to detect antigens that are delivered in that manner. But the thing that didn't make sense to me is that you can also take fish and you can immunize them by injection. You can go retroorbital into the bloodstream, you can go intramuscular, or you can go IP, and all of these elicit robust adaptive immune response. And this intramuscular one actually really bothered me because if all these fish have is these lymphocytes around their gills, nasopharynx, gut, and spleen, who is detecting that intramuscular antigen? And so I started to suspect something else might be going on, and it's something that I wanted to study in zebrafish. So for those of you that are familiar with zebrafish, most of the work done in zebrafish is done in larval zebrafish. But the issue from my perspective is that zebrafish don't really gain an adaptive immune system until about 6 weeks of age. This is well after most people are studying these animals. And one of the problems with studying these animals as they get older is that they gain their namesake stripes. They become pigmented, which makes them much harder to do live imaging on. Fortunately for me, a number of years ago, a laboratory made this mutant line called casper. It's got 2 genes that are mutated that disrupt the pigmentation. And so now you've got this optical transparency that is carried into adulthood, allowing us to do some high resolution imaging in adults. And we're going to be using a lot throughout this talk these LCK GFP zebrafish. So these are going to be zebrafish where GFP is driven by a T cell specific promoter. So this was really the first image that I took when I came to the Huttenlocher lab, which I basically I said, let's just look and see where T cells are across the body of this animal. So this is looking at an entire 10-week-old zebrafish. When you see this image, you're going to notice a couple of things right off the bat. It's got this very bright spot near the head. This is the thymus, the organ where these T cells are being generated. But the other thing that you're going to notice here is that they've got this zigzagging organization of green spanning the entire body, particularly localized on the dorsal and ventralmost portions of these animals. Here's an inset here where you can really see this zigzag pattern more clearly. So this was strange and not something that we expected to see and not something that we had ever seen reported before. As I mentioned, there had been a lot of work that was done on zebrafish in larvae. And so we wondered, is this something that's also present in larvae, or where does this structure show up? So we looked at the zebrafish across development, looking at larval, juvenile, and adult stages. What you can see here is that you don't have this network in larvae. Around 7 weeks of age, you start to see this network showing up. By 10 weeks of age, it's very prominent, and this is maintained into adulthood. Now, if you were to zoom in on these larval zebrafish, you would see individual T cells, but they're not organizing into this higher order structure. So our laboratory, I'm really the only T cell person in our laboratory. Most of us in the Hutton Locker lab study neutrophils and macrophages. And so I wondered, is this zigzagging network something that is unique to T cells, or are other leukocytes organizing into this structure? And what I hope you can see here, these are T cells in green, macrophages in red, neutrophils in blue. What I hope you can see here is that only T cells, amongst these leukocytes at least, only T cells are organizing into this zigzagging structure. So at this point, we decided, well, we better name this thing. This was the only thing that I've ever named so far in biology, so I wanted to take it seriously. So we thought there was two aspects of this that we wanted to capture in the name. The first is that it has this zigzagging, regularly repeating pattern. In geometry, that's often referred to as a tessellation. A honeycomb, for instance, is a tessellation of hexagons. And then primarily what I just showed you is that it seems to primarily be lymphocytes, at least T cells, that are in this network. So we called it the tessellated lymphoid network, or TLN. Now, one of the things that we noticed when we were doing this, that you probably noticed too, is that this zigzagging nature of the T cells looks fairly reminiscent of scales. So this is the image that I started with, and then this is beautiful image from the Traver lab or from the University of Virginia, excuse me, showing the scales. You can see that there's some kind of a pattern here, and they look like they might be associated with one another. And so I took my LCK GFP zebrafish and I stained them with a dye called alizarin red, which is going to bind to calcified extracellular matrix. And what I hope you can appreciate here is that there's a very clear correlation between the TLN and between the scales. Everywhere that the TLN is zigging and zagging corresponds perfectly to where a scale is in this body. And in this orthogonal projection, you can see that both the TLN and the scales are very superficial structures on the fish. Now, if we really zoom in on the very dorsalmost portion of these animals, where I've got these scales that are pseudocolored here in yellow, what I hope you can appreciate here is that what's going on here is that the scales are coming out of the body in an overlapping fashion, and the TLN is localized right into that pocket formed by those overlapping scales. You can really appreciate that in these alizarin red stains, where you can see that there is a scale both above and below where the TLN actually is. So it's underneath the scale and it's above the base of the subsequent scale. And that's sort of the tissue organization that's going on here. So I wanted to be able to get these cells out to study them. Now the problem here is that this was a very diffuse kind of tissue. It wasn't like an organ or a lymph node that I could just pluck out and mash to get a single cell suspension. But because this was so intimately associated with the scales, we said, well, what happens if we descale these fish? So if there's any people that are like to fish, or you like to buy whole fish at the grocery store, you would have done something very similar to this, except instead of using a knife, I'm using a very small scalpel. Basically, we take these euthanized fish and we descale by scraping against the grain with a scalpel. All the scales come off. And what you notice here is that when you get rid of the scales, the TLN goes with it. Now, this isn't just because I'm pulling the wool over your eyes and this fish never had T cells. You can see in the nearby fins, the T cells are still there. Now, if we do this descaling protocol in buffer, so we basically take the euthanized zebrafish, put it in PBS, and then do our descaling underneath that buffer, what we find is that we immediately get this single cell suspension consisting of GFP positive T cells, as well as a mixture of a bunch of other cells. So now we have the ability to actually get these cells out. And if you're trained as an immunologist like I am, and you get a single cell suspension, your instincts kick in and you say, I better do some flow cytometry. So that's what we decided to do here. So in addition to just simply doing flow on these cells, I wanted to compare the TLN to all of the other known reservoirs of T cells in the fish. So we pulled out the TLN, the gills, the gut, the kidney, and the spleen, and we prepared single cell suspensions from every one of these sites. Here I'm using dual reporter LCK GFP CD4 M cherry. I'm sure most of you know that helper T cells are CD4 positive, so it just gives us another readout of T cells here. And what you can appreciate is that about 50% of the cells, total cells that we're pulling out of the TLN are T cells, and a number of them are CD4 positive. If we simply count the number of live cells coming out here, you can see we're pulling out a lot of cells from the TLN, more than most sites except for the kidney. But if we specify the GFP positive cells, you can see that we're pulling out about 100,000 T cells from the TLN, more than all of these other sites, actually more than all of these other sites combined. So this is not some minor or supplementary population of T cells. This is the largest single reservoir of T cells in the entire fish. So at this point, we decided we should probably do some single cell RNA sequencing to understand, in addition to T cells, what else is here and what are the kinds of T cells that are here. So this was a nice sanity check for us because about half the cells that we found in our single cell RNA sequencing were T cells matching our flow cytometry. We found a number of other leukocyte populations. We found macrophages. We found a small number of B cells, a small number of dendritic-like cells. And then in addition to that, we also found a number of a number of non-leukocytes, things like epithelial cells that you would expect to come out when you're scraping the skin of these animals. A lot of these T cells that we pulled out, if we further subdivided on them, expressed markers of naive T cells. They highly express CCR7 and CCR9. These are the big chemokine receptors for naive T cells in mammals. They didn't really express any cytokines or things that you would associate with an activated or with a memory T cell that really seem to be naive-like in their nature. Now, in addition to these T cells, we wanted to focus on the big thing that causes a lymph node to become organized into a structure is the production of chemokines and the associated expression of chemokine receptors by leukocytes. And so we wanted to home in on the chemokine receptors and the chemokines. What you can appreciate here is that this is identical to mammals. We see CXCR4 expression broadly expressed by most of the leukocytes that we identified in this population. CCR7 is more or less exclusive to T cells. And again, this is exactly what you see in a mouse or in a human. And then some of these other chemokine receptors were broadly expressed. We found this population of intermediate epithelial cells that was expressing a lot of the chemokines for these receptors. I'm not going to talk any further about that, but I'm happy to talk about it in the roundtable. We're trying to hone in on this population as a major organizing population of the tessellated lymphoid network. So summary number one of what I've told you so far. T cells organize into a tessellated lymphoid network in adult zebrafish. The TLN is the largest reservoir of T cells in the host, and naive T cells and antigen-presenting cells are abundant in this network, just like they would be in a mammalian lymph node. So like I talked about at the beginning, the reason why the lymph node system works isn't just because you've got these bags of lymphocytes around your body. It works because T cells are constantly moving between those lymph nodes. So we wanted to understand whether these T cells in the tessellated lymphoid network were also motile and what that could tell us about antigen search in these fish. So the first thing that we noticed when we did live imaging of these cells is that they appeared extremely ballistic in the way that they migrated. These T cells tended to pick a direction, and all of them collectively migrate in that same direction. They migrated at about 14 microns per minute, fairly fast. This is actually dead on what you find in a mammalian lymph node. But again, their direction was very high, very close to 1. These cells pick a direction, and they migrate in that direction. Now, what we noticed for these T cells that were doing this migration in the TLN is that they tended to form these head-to-tail interactions, where you can see the leading edge of one T cell latching onto the rear or the uropod of the T cell that was in front of it. And the way that this played out in terms of migration is that these T cells tended to migrate as these little multicellular units rather than as individual cells. Now, this movie here is not particularly impressive. I'm deliberately showing you one where the TLN has not fully formed so that you can see and appreciate individual T cells a little bit better. But by the time you get to full adulthood, you've got a really amazing situation going on here, where these T cells are utilizing this TLN to basically zigzag their way in a collective stream through the body. They're all moving more or less the same direction. Some of them will leave and come back, but you can see that the overwhelming majority of cells are migrating in this remarkably coordinated fashion. And the thing that was cool about this is that the direction that these cells migrate depends on where you look in the body. So if you look on the dorsal side of these animals, these T cells are more or less all migrating from the tail towards the head. But if you look on the ventral side of the body, the opposite is true. They're migrating from the head towards the tail. I'm not going to show this data, but we also show in the supplement of the paper that I sent that these T cells will move from the ventral side to the dorsal side at the base of the caudal fin. So when you piece all of this together, more or less what you find here is that these T cells are utilizing the TLN to move in a loop, a coordinated dorsal to ventral loop or ventral to dorsal loop around the body. So this migration was so organized that we actually originally thought that these cells must be within some kind of a vessel. We got the lymphatic vessel. So this is a lymphoid tissue. Lymphatics are the actual vessels that collect lymph. Fish do not have lymph nodes. They do have lymphatics. But what we found here is that there was no overlap between the lymphatics and the tessellated lymphoid network. So when you're looking at the zigzagging structure, you can see that there are some places in a Z projection where the lymphatics and the TLN will overlap. But if you actually look in the orthogonal view, the lymphatics are deeper in the tissue. So the TLN is not simply within a lymphatic vessel. We saw the exact same thing to be true with the blood. These are also not in the blood. So they seem to be migrating and they don't seem to actually be in a vessel. Now, one of the things that we noticed, especially for me coming from the mouse lymph node world, is that these T cells are migrating very directionally, and that is very different from how T cells migrate in a lymph node. Because T cell migration in a lymph node has been very well characterized. It's typically described as a random walk. Basically, this mode of migration that allows T cells to scan as many APCs in a given amount of time to effectively find antigen. That's very clearly not what the T cells in the TLN are doing. And in fact, we can visualize our T cells and APCs like macrophages at the same time. And what I hope you can appreciate here is that these T cells are just blowing past these macrophages, touching them transiently, if at all. And so we began to suspect that this mode of migration might not actually be particularly good for searching for antigen, but we wondered if the behavior of these T cells might change in an environment where antigen was much more likely to be found, namely an infection. So I had to develop, nobody had done these adult infections before, so I developed an infection model. Our lab has used Listeria monocytogenes to infect the skin of larval zebrafish, and so I started working with that. Basically, what I'm doing here is I'm inducing a puncture wound with a needle. I'm then putting these fish into a bath with fluorescent Listeria for 2 hours, and then we're moving them into fresh water until we're ready to use them for our experiments. What you can see here is that this Listeria, which we can track in red, grows over about 1 to 2 days and then is present up until about 5 days. And by 7 days post-infection, this is cleared. You can see that the TLN very clearly sort of changes in response to this infection. And particularly between 3 and 5 days, you get what looks like accumulation, maybe even proliferation of T cells in this nearby tessellated lymphoid network. So we decided to really zoom in on these T cells in these infection proximal regions. So at this sort of higher magnification, better resolution imaging, you can see that there are these aggregates of Listeria, and there are T cells in proximity to that red Listeria here. So we did live imaging to understand whether the behavior of the T cells changes in the areas around the infection compared to the homeostatic TLN. So I've got 2 movies here. This is showing you a TLN in a control fish where these T cells are doing this collective streaming like you just saw. But if we look at these T cells that are in proximity to the infection, you can see their behavior changes just dramatically. They stop doing this collective streaming behavior, and these T cells start migrating, first of all, individually, but undergoing much more of a random kind of walk. One of the ways that we can quantify this behavior is with directionality. How straight are these cells moving? Again, in a control TLN, these cells are moving very straight, approaching 1. This drops significantly in the regions around the infection in an infected fish. And this is a local phenomenon. So if we look in an infected fish, but far away from the site of the infection, these T cells maintain this very directional mode of migration. So something about being in proximity to this infection or this tissue damage triggers a switch in the way that these T cells are migrating. And one of the cool things that we can see here, this is not the highest resolution image, but what you can see here, I've got this T cell marked here with this dot, and I've got 3 macrophages here. You can see that when these T cells transition into this random walk, it allows them to sequentially interact with antigen-presenting cells in the same kind of way that T cells are known to sequentially interact with antigen-presenting cells in mammalian lymph nodes. So all of a sudden, you've got a situation that looks like antigen search in a lymph node when you have an infection. So that was looking at 2 days post-infection. By the time that you get to 4 days post-infection, T cells have begun to form these very stable interactions between macrophages that are proximal to the infection site. You can see here these macrophages just wrapping around these T cells. We decided to do single-cell RNA sequencing of these T cells that are in this infectional proximal region and compare it to a control fish to understand, are there any indications that T cells are actually detecting antigen and activating within the tessellated lymphoid network like they do in a mammalian lymph node? So first things first, we can group these T cells into different subgroups. We find that the naive-like proportion of T cells goes down in the infected group or in the infected fish, and the proportion of cycling, proliferating T cells goes up. This is exactly what you would expect if T cells are beginning to activate and proliferate in response to antigen derived from this infection. And we also see very clear signs of activation from T cells that are isolated from the TLN. So this is a big type 1 type of infection. So interferon gamma is the big cytokine that you would expect T cells to produce in response to this infection. And lo and behold, we see a whole bunch of T cells beginning to express interferon gamma in the TLN. So the major points that I want to sort of convey to you about this story, front to back here, is that T cells organize into a tessellated lymphoid network during the juvenile stage of zebrafish, and this is maintained into adulthood. This network is the largest reservoir T cells in the entire host. T cells utilize an extremely coordinated ballistic movement to traffic through the host in a ventral to dorsal loop. In inflamed regions, T cells transition to a random walk and form stable contacts with antigen-presenting cells. And T cells activate and differentiate within the TLN during infections, just like they would in a mammalian lymph node. So I think as most of you know, we published this in PNAS, and the nice thing about the TLN is that it's beautiful. And so we were fortunate to get one of our pictures on the cover of the issue that we were in. I'm going to show you a couple slides of data that we've done since this publication. I'm keeping most of it out because many of you brought up the things that we're interested in your excellent questions. But I'll tell you one very short story about something else that we noticed. So when I was working on the tessellated lymphoid network story, one of the things that we always notice is that you've got the tessellated lymphoid network in the pocket formed by overlapping scales, but you've also clearly got T cells that are on the outside of this pocket, or basically on the outermost epidermis of the skin. So we wanted to know whether these represented different populations of T cells from the TLN. We did high magnification imaging of these cells. We noticed a couple of things right off the bat. First and foremost, morphologically, they look very different. These T cells that are on the outer scale have a very dendritic or stellate kind of morphology. And this is very different from the compact morphology that you see in the TLN. These T cells in the scale, they don't express CD4 to the degree that the TLN T cells do, but they do form a network with CD4 positive macrophages that you can see very, very nicely here. Now, these CD4 positive macrophages have already been described as being equivalent to Langerhans cells, these tissue resident macrophages that are in the skin of mice as well as ourselves. You can see here these T cells forming this network with this population of Langerhans cells. Now, one other distinction between the scale and TLN T cells is that scale T cells do not migrate. Under homeostatic conditions, they are very sessile, and this differentiates them from the TLN T cells. So when I was taking these images, having come from the mouse world, one of the things I noticed was that these T cells and this network that they're forming with Langerhans cells looks very similar to a population of T cells that's in the skin of mice and to a lesser extent in the skin of humans, called dendritic epidermal T cells. They also have this dendritic morphology. They also form this network with Langerhans cells shown in red here. So we began to suspect that maybe these are a homologous population that are also present in zebrafish. So one of the things that these dendritic epidermal T cells do in mice is that they respond to tissue damage by rounding up, losing this dendritic morphology. So I introduced a puncture wound in the region near some of these cells, and what you can see is you get this clear loss of dendritic morphology in the regions around the puncture that is not present in T cells that are distant from the puncture. These cells are behaving almost identically to these dendritic epidermal T cells in mice. Now, one of the cool things that we can do in zebrafish that you can't do in mice is that zebrafish develop externally. And one of the things that we know about these dendritic epidermal T cells is that these are the very first T cells to ever leave your thymus. So during embryonic development, these are the first T cells out of the developing thymus, and they immediately traffic to the epidermis and embed themselves in that site. And so we wanted to know, can we watch this process happen in real time? So I'm showing you here a blood vessel reporter with the LCK GFP. And the blood vessel is nice here because the thymus forms right around these arches formed by the developing gills. You can see maybe at 3 days post-fertilization, you've got some indication of a thymus. By 5 days post-fertilization, the thymus is now formed and present. And right around the time that the thymus shows up, we begin to see T cells adhering to the blood vessel right next to the skin in these larval zebrafish. Now, we can watch these fish as they develop going from 12, 17, 22 to 27 days post-fertilization. And what you can hopefully appreciate here is that over these stages of larval development, these T cells will leave this blood vessel and progressively spread out across the skin, such that by the end of larval development, you've got this network already formed of these T cells and Langerhans cells shown here in magenta. And these T cells are extremely motile early during this phase. You can see here with these cell tracks and the associated velocity here. But by the time you get to the end of larval development, around 27 days post-fertilization, they have completely stopped. And part of the reason why I did this project is because despite the fact that fish do not have an adaptive immune system until they're about 6 weeks old, people study T cells in larval zebrafish, and they will tell you that they're searching for antigen. And I'm here to tell you that they're not. They're colonizing these tissues. That's why they're migrating. They're not searching for antigen. If they were searching for antigen, they wouldn't stop, at some point during development. But knowing that these T cells are related to these epidermal T cells in mice gives us a better understanding of what's going on in larvae. And I hope that people in my field stop looking at T cells in larvae as though they were the equivalent to a T cell in a mammalian lymph node. That is the end of my soapbox on that. With that, I will say thank you to people in my lab that were critical for this. Anna is a great mentor. I actually had never seen a zebrafish when I came here to do my postdoc, and so Julie was the person that trained me on the zebrafish. Simone helped with the listeria infections. John Schroep was helpful at doing the quantification of the cell migration. And then the big one, Yaron, did all of the single cell RNA sequencing and associated analysis, and I don't know how to do any of that, so without her, we literally couldn't have done any of this. Thanks to our collaborators and to the Gene Expression Center. And with that, I'm going to finish on a stupid pun. I don't know how the round table works, but
Right?
Yep.
They're not constrained in the,
yeah.
construct that you looked at, they're nearby. What's making that pattern?
Yeah, we don't know. I mean, the zigzagging pattern, I think, just comes from the fact that they're in that scale pocket. Why they stream, we don't know. I have, so one of the things I didn't show here is that you don't see a lot of other leukocytes in the TLN, but the ones that are in there do not stream. And so it seems to be a T cell specific thing to do this sort of streaming collective motility. They have a chemokine receptor called CCR7 that none of these other cells have. And so I have made the knockout of it. I have a hat now, but I don't know what the full knockout looks like. We think that might be involved. But that could maybe explain a little bit of the streaming. The thing that's confusing is that the only thing that looks anything like this is dictyostelium. I don't know if you guys are familiar with dictyostelium. It's a soil amoeba that does this cool streaming migration that looks a lot like this. And the way that they do that is that the cells simultaneously sense and produce a chemoattractant. So they produce the chemoattractant in the rear, and that helps them form these sort of head to tail interactions. We think something similar might be going on here, but it is early days on that. Yeah. Mhm. Yeah, so one of the things that came out of the single cell data is this population of epithelial cells that produce the chemokines that we know T cells like. And so this is like the most disappointed I've ever been in an experiment. So we thought if we labeled these cells that we would probably see a similar zigzagging pattern, and the T cells are migrating on, like those cells are producing these chemokines and also providing a scaffold for those T cells to migrate. So I got really excited because we made that reporter line and it forms a zigzagging network just like the TLN, but they're slightly offset from one another. The T cells don't actually migrate on those cells, and that has been confusing for us so far. I don't know what they're migrating on. I can tell you a bunch of stuff that they're not migrating on, but what they're migrating on, we don't know so far. It's been a big question for us. We're hoping that if we get rid of CCR7 and this goes away, that'll indicate something about CCR7 is critical. But right now, it's a big question.
Not only were you
Yeah.
what do you think build it right there?
So this has been a big question for us. So yeah, you could think about it sort of large scale. So like when you get the evolution of amphibians and then the reptiles and birds, most of them don't have lymph nodes. Do they have a TLN or do they have a TLN-like structure? We have no idea. Certainly has never been described. The more interesting and tangible question that we're getting at right now is what happens when you have a fish that you've either mutated it such that it no longer has scales because those exist, or a fish that naturally lacks scales. So for instance, catfish all lack scales. So that actually has been, I have that in here. That has been one of the first hits here, is that we got all these mutant lines that are involved in skin appendage formation. I only have this one that is a mutant in fibroblastic growth factor. These are called Haggeroma mutants. And one of the cool things about these fish is that the scales form, but they're really disorganized. Their development is a little bit off. But the surprising thing is that these fish 100% lack a TLN. They've got T cells in other tissues. They've got a thymus, but they are completely missing a TLN. So now we're very interested in the signaling pathways that drive scale formation. We think that's being co-opted by the TLN to help this structure develop. So we've got a couple other mutants. I don't have the T cell reporter on the background yet, but we've got these EDA knockouts that are like absolutely critical for scale formation. And then we've also developed the ability to section and stain for the TLN in wild, like normal fish. So we're trying to get our hands on, let's say, trout and goldfish and catfish to see, first of all, how broad is this network? And if you have a fish that is lacking scales, does it have anything that looks like this? We think it probably does not. And that's actually interesting because catfish, I'm talking a lot about this, but catfish are highly susceptible to bacterial infections. It's a huge problem in their skin specifically. It's a huge problem on fish farms. And so it'd be cool if the reason for that is that they're missing this lymphoid tissue. Yeah, I think of the fact that it's not in the middle. It's kind of like how if you've got two lanes of cars on a highway, you want a median in between that. I think if it was kind of everywhere, then the ability to do the streaming migration would be messed up. But because we don't understand what's guiding the, like, in addition to the streaming, why does it always go in this counterclockwise loop? We don't understand that either. And so, yeah, that's another one. But it makes sense to me that they would have evolved to have it preferentially on both poles so that they're not kind of getting mixed up. And because presumably there's like two signals guiding that direction, you probably don't want them mixing with each other for this to be an efficient system. But it's not just like an artifact of the microscope. They truly are not in the middle of the fish. Mhm. Yeah, so where the crossover happens is actually like where their biggest you can cut off the larval zebrafish tail. In the adults, it's like where their biggest artery loops, and so it bleeds out rapidly if you try to do that. There are other things, so you can pluck some scales, and that can kind of disrupt it in a way, but we haven't really pursued that fully yet. We're hoping that some of these scale mutants will help us figure out some of those things. We're also working on doing some just laser ablating a region and seeing how the T cells react to that. But yeah, I don't have any answers so far. Yeah. Yeah, yeah, it's a good question. So the reason why I did it slightly more dorsal is because there is a major blood vessel right in the middle. And it's like when I do my puncture, I killed several of the fish because you hit that artery. So I did it more dorsal. If you do it more ventral and also miss that, then the ventral ones do go there. It seems to be whichever one is closest. And you actually often see, let me see if I can see this. Sure. You kind of often see what looks like it almost coming at it from both directions a little bit. This one is an infection that's very much more dorsal, and you're getting what seems like the dorsal TLN being the primary thing that's reacting. But you kind of see a little bit of accumulation in the same plane, but on the ventral side. I think probably whichever's closest is what works, but they probably all are aware of the fact that there's an infection nearby. Yeah. Yeah, I think it's because T cells, myeloid cells in general don't have the antigen receptor diversity problem that T cells and B cells do. And so if you've got an infection in your skin, I mean, people in my lab would dispute this because there are neutrophil subsets, don't get me wrong, but like a neutrophil is a neutrophil. So like it doesn't matter if it's that neutrophil over there or that neutrophil over there that sees that bacteria, they're both going to do the same job. Whereas because T cells all have different antigen receptors, I think that is what necessitates this higher order organization. Because two T cells reacting to an antigen, one of them might be specific for that, the other one might not be. So it actually matters how these T cells are moving around the body in a way that just simply doesn't for myeloid cells. And it's not that they're, the macrophages are organized in the sense they form these nice layers on the outside of the fish, but yeah, they don't need to move in the same way because they all express germline encoded receptors, whereas T cells have this VDJ recombination that gives them diversity. Makes sense. Mm. Mhm. Yeah. It's a good question. One of the things we noticed, and I didn't put it in the paper, is that when you've got a situation like this going on, the T cells that are here end up migrating very, very slowly. It's almost like there's like a log jam kind of thing going on. Yeah, so I bet that there probably would be an issue with finding antigen. It would actually be interesting to do like a puncture wound, let's say here and a puncture wound here, and try to give them two different pathogens to see, does that really affect their ability to clear it? It definitely it is like a single lane highway. So when you mess it up, it does seem to be like it gets backed up, and it's kind of funny actually to watch that they all get jammed up there. Um, but yeah, I mean, this is the system they evolved to have. It doesn't seem perfect, but it's what they're doing. Thanks. Thanks.